Imagine being able to rewrite the code of life itself, correcting genetic defects, curing diseases, and even preventing the extinction of species. This is the power of gene editing. Gene editing is a revolutionary biotechnology that allows scientists to precisely alter the DNA within living organisms. Using tools like CRISPR-Cas9, researchers can cut, insert, or modify genes with incredible accuracy, opening up possibilities that were once thought impossible. So, why is gene editing so important? The potential for gene editing is immense. It provides a solution to genetic disorders like cystic fibrosis and sickle cell anemia, diseases that once had no cure. By editing genes, we can treat or even eliminate these conditions at their source. Gene editing is also a game changer in agriculture, enabling us to create crops that are more resilient to climate change, pests, and disease, thus addressing global food security. On top of that, it offers possibilities for saving endangered species and even bringing extinct animals back to life. But how does gene editing work? At the heart of modern gene editing is a technology called CRISPR-Cas9. CRISPR is a natural system found in bacteria that acts as an immune defense. Scientists adapted this mechanism to target specific DNA sequences in other organisms. The Cas9 enzyme acts as molecular scissors, cutting the DNA at a precise location, and guide RNA directs it to the exact spot where changes need to be made. This allows for highly targeted modifications, whether it's fixing a genetic mutation or inserting a beneficial gene. Now, let's take a look at how gene editing has evolved over time. Gene editing began as a concept in the 1970s with the first experiments in recombinant DNA technology, which allowed scientists to manipulate genes. However, the real breakthrough came in 1993 when researchers developed the first artificial zinc finger nucleases, a tool for cutting DNA. These tools were powerful but still difficult to use and expensive. The game changer came in 2012 when Jennifer Doudna and Emmanuelle Charpentier discovered CRISPR-Cas9, a tool that was simple, cheaper, and far more efficient. Their discovery made gene editing more accessible, and by 2015, the first successful gene-edited animals were born using CRISPR. Since then, gene editing has advanced rapidly. In 2017, Chinese scientists used CRISPR to edit human embryos, and by 2020, gene editing therapies were used to treat genetic disorders like sickle cell anemia. Today, gene editing is used in agriculture to create drought-resistant crops and in medicine for clinical trials focused on curing genetic diseases. Looking ahead, the future of gene editing is full of promise. In recent years, new technologies like base editing and prime editing have been developed to make gene editing even more precise, reducing off-target effects. These innovations could bring about new treatments for diseases like cancer, HIV, and even genetic aging. As we continue to refine gene editing, the possibilities are endless. Imagine a future where genetic diseases are eradicated, crops grow in extreme environments, and endangered species are safe from extinction. Gene editing is reshaping our world in ways we never thought possible. From curing diseases to enhancing crops, the future of gene editing holds limitless potential. But as we continue to explore this frontier, it's vital that we move forward thoughtfully, balancing innovation with ethical considerations. Gene editing isn't just a technology, it's the future of life itself.